It's an honor to be here. So my company was just incorporated this summer, so it's a very new company, just a couple months old. And the main problem that we're focusing on is the blood-brain barrier. As most of you might know, majority of therapeutics just simply cannot cross the blood-brain barrier, and this poses a significant issue for trying to target therapeutics to the brain for treating any brain diseases. So even in investigational therapies, in the drug discovery process, even investigational therapies that looked kind of promising or quite promising in preclinical studies often fail to successfully translate into human use. And this is where we position our company. We're here to help pharmaceutical companies identify what is more likely, what therapies are more likely to fail or what are more likely to succeed. And this could end up potentially saving pharmaceutical companies billions of dollars down the road to focus in and narrow in on the drugs that are most likely to succeed and avoid drugs that are least likely to succeed in humans. So the technology that we are working on and that we have is the human blood-brain barrier organoids or assembloid models. This is something that we have worked on for more than a decade, for more than 10 years in validating it to show that it can very faithfully recapitulate the functions and biology of the human blood-brain barrier in vivo, as well as validated a lot of our studies in mice as well. And we've shown that our studies, our platform is very reproducible and can help us delineate can help us predict what is likely to be permeable or non-permeable. Our platform is very modular. So that means that we can incorporate even patient-specific stem cells using patient-specific BBB stem cells, and we can even incorporate disease modeling, such as including patient-derived brain tumor cells, for example. And this makes it more feasible for us to conduct personalized medicine or facilitate personalized medicine in the future. We can also include immune cells to make it even more physiologically relevant. And as I've mentioned, we have been able to show that our system is very predictive, and we can scale it up also to high throughput capacity. So more than 50% of current studies still employ legacy BBB models, such as the Transwell model. And the Transwell model has been around for a very long time. It's very familiar, and so it's still pretty widely used. So we decided to do a side-by-side comparison between our BBB assembloid and the Transwell model and measure its ability to predict permeability of certain compounds. So the question here is quite simple. We have a positive control BBB penetrant compound and a control non-penetrant compound, and we wanted to see if we can cross the barrier. And so when we did our study, what we show is that, in fact, the BBB penetrant peptide in red here can, as a matter of fact, cross the barrier in the Transwell. But what is also a very well-known problematic issue with the Transwell model is that it tends to be quite leaky. And indeed, that's what we saw in our control non-penetrant peptide. We also saw an inherently high level of leakiness, even after 40 hours of investigation. So when we compare it with our BBB assembloid model, this is what it looks like. The surface of these assembloids are what constitutes the barrier, serve as the BBB, with a high level of BBB function. And when we put the BBB penetrant peptide and the non-penetrant peptide together with our assembloids, this is indeed, we saw within 3 hours there's a significant difference in permeability level that we can differentiate, where we saw the BBB penetrant peptide can in fact cross very, very highly, but the non-penetrant peptide was excluded and cannot cross the barrier. And this is something that we can observe even within 3 hours, the high level of differentiation between the two compounds that was not being able to detect by the Transwell. And when we took a closer look as to why this might be the case, we saw that indeed the BBB assembloid platform has a much higher level of these BBB markers that are critical to confer the function and the biology of the blood-brain barrier compared to what is being expressed on the Transwell model. This is just a quick comparison for showing our BBB assembler and how it compared to current gold standard assays. You can see that our BBB assembloids really does hit a lot of the key metrics of what is important for assessing a BBB assay, such as it includes barrier awareness and has higher predictive power to predict human BBB permeability. We've also been able to show that we can work with this platform at a high throughput capacity, and we have been able to show very good reproducibility with our platform. And you can see that compared to animal models, our cost of operation is actually significantly lower. What we have recently been doing, and I'm showing you here, is that we have also been able to incorporate disease modeling in our platform. And in this case, I'm showing the incorporation of glioblastoma cells, or very malignant brain tumor cells, in our BBB assembloids. So on the left here, in blue, I'm showing a primary GBM model, and on the right, it's a recurrent GBM model. And the primary responds very well to standard of care TMZ, and the recurrent, this is a recurrent model coming from the same patient, just at different disease stage. You can see here that the primary tumor model responds very well after 9 days of temozolomide treatment. We can see a significant reduction in the primary tumor in our BTB assembloid. But on the right, with the recurrent model, there is no response to TMZ. And so what we're showing here is that we can actually model the standard of care response, at least as a validation, in a barrier awareness setting. And also, this platform allows us to ensure that this dosing regimen and this concentration of drug that we're using is healthy and non-toxic, is non-toxic to healthy BBB cells. So the next thing that we really want to do in the company is to leverage our assembloid model and build this next generation BBB predictor calculator, which will be powered by AI. And the first thing that we need to do is to generate a big data moat that we can continually add on as we validate more and more clinically validated compounds. Then we will want to integrate machine learning and use our model to sort of train this platform and build this BBB calculator where pharmaceutical companies or anyone who's interested in drug development to the CNS can just upload their structures, and we can then calculate and generate a number or a likelihood. How likely is your compound going to cross the BBB, or how unlikely it is to cross the BBB, or maybe it's just sort of in the middle, and what you can do to sort of make it more likely to cross the blood-brain barrier. So why is this important now? The NIH has recently had a significant policy shift thanks to the FDA Modernization Act to push for more human-relevant models and reduce animal model use. And the push for human-relevant new approach methodologies is really something that the company working on BBB assembloids is really heavily focusing on. Pharmaceutical companies are also spending a lot of money on AI-integrated platforms. Until today, currently there's no BBB predictor tool that exists that is powered by AI, so we would really be interested in doing this. So the company is interested in expanding our data moat by up to more than 1,000 compounds to use it to train and build our AI BBB calculator. And we would like to launch this when we have it at pilot studies with a few pharmaceutical companies and continue to build on that from there on. We're exploring opportunities for partnerships and funding, and I welcome any discussion for collaboration. So thank you very much. I'll leave it at that.